Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Aging is inevitable, and goes along with many changes in cells, tissues, and organs, including DNA damage, mitochondrial dysfunction, and telomere loss. But why we age in the first place and what drives these changes is still unknown. As cells age, the architecture of chromatin, which packages DNA, unravels. As cells age, the nuclear lamina weakens and frees the heterochromatin, which loosens, allowing previously silenced genes to be expressed. Previously, Beck and his colleagues showed that heterochromatin formation only regulates the expression of CGI genes, while CGI plus genes are silenced through another mechanism called repressive polycomb bodies. In the new work, our hypothesis was that aging, and its associated chromatin architecture disorganization, results in dysregulation of genes lacking CPG islands, says Beck. In some mice, CGI genes in the kidneys were upregulated, while other mice of the same chronological age didn't show this misexpression. When the researchers took a closer look, they found that mice with upregulated CGI genes had a higher incidence of renal dysfunction. Looking further into the link between chromatin architecture and CGI gene misexpression, the researchers turned to a receptor called lamin B that tethers heterochromatin to the nuclear envelope. In mice with a non-functional lamin B receptor, they observed looser heterochromatin and CGI gene misexpression. In other words, nuclear architecture disruption and heterochromatin decondensation lead to CGI upregulation, says Beck. The team is investigating whether it also works the other way around, with upregulation of CGI genes causing or facilitating chromatin decondensation. This is a strong descriptive study showing an association between heterochromatin disruption and the activation of genes devoid in CGI promoters, CGI, writes University of Edinburgh geneticist Tamir Chandra, who was not involved in the study, in an email to the scientist. Our hypothesis was that aging, and its associated chromatin architecture disorganization, results in dysregulation of genes lacking CPG islands. Unexpectedly, in mouse cells, CGI genes located within heterochromatic domains were rarely misexpressed during aging, while CGI genes forming local heterochromatin within largely euchromatic domains were overexpressed, Beck adds. Accordingly, when they looked for sites where transcription factors might bind in euchromatic and heterochromatic domains, they found that CGI genes within euchromatic domains have more transcription factor binding sites compared to CGI genes within heterochromatic domains. Additionally, CGI genes within euchromatic domains that are upregulated during aging contain more binding sites than genes that are not upregulated. Further investigation as to why it is not CGI genes located in heterochromatin that are affected by the disruption would be interesting, writes Chandra. The researchers also investigated whether CGI genes are connected to what's known as cellular identity. Cells making up the heart, muscles, kidneys, or other organs usually express different genes to carry out their functions. As cells age, they also lose this cellular identity, and the researchers wondered if misexpression of CGI genes could help explain why. That is one way how aged cells lose their identity, suggests Beck. Aged cells also secrete signals in an uncontrolled way, but what triggers this secretion is not yet known. Analysis of the products of CGI genes in mouse kidneys and hearts indicated that many encode secreted proteins, including cytokines, chemokines, growth factors, and proteases. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.